Hello and welcome back to my channel. This is your tutor doctor from DB Tuitions. I hope everyone is doing good, and today we are going to break down question 5 of paper 1 AQA GCSE biology, higher triple science. Now, this question is based on osmosis required practical, and I want you guys, all my fams, listen carefully, because this is one of those questions where you found, all of you, you know the topic, but you still lose marks because you don't want to write the method, explanation, key terms, exactly the way the examiners want. So I'm going to break it down step by step, and I'm going to show you what is going to earn marks and what doesn't. And I'll give you some memory tricks along the way, so this becomes a full score question for you in the exam. Now, if you haven't already started following me, I would strongly advise do that, because I don't do hours together, boring, lengthy videos of whole paper. I always say, let's conquer one question at a time, slowly, and I'm going to make all of the question, past paper questions, question by question breakdown, teaching you topic little by little. Even if you start now in December, you will still be guaranteed a grade 7 if you follow my revision plan, which I've also made a video about. If you can go through my videos, you will follow the 5-point plan for your GCSE A-level, no matter what academic exams you're doing. So without much ado, let's move on to question 5.1, and as I always say, this is a 6 marker. So 6 markers are important. They make or break your grade, so it's very important that you read the question and follow what's required, and do not overly write. Remember, always write in paragraphs, use all of the space available, do not bullet point your 6 markers unless specifically told you so in the question. Now, the question says a student investigated the effect of concentration of salt solution on the mass of uncooked potato pieces. There is a graph given here which says figure 4. Now, what I want you to understand, some of you are very good in interpretation of graphs, and some of you, if you're anything like me, like when I was in my GCSEs, I used to struggle with the graphs. Now, typical graphs are given in this fashion. So you have your y-axis, you have your x-axis, and there is some kind of a figure. But remember, not always. If your y-axis is taking negative values, the graph can be given like this. So that is your positive y-axis, this is your negative y-axis, and as always, x-axis. Now, in this experiment, you're taking salt solutions of varying concentration, and you're taking potato pieces, keeping them for some time in each salt solution of certain strength of concentration, and then measuring the mass before putting them and then after. And that is why on the y-axis you're taking mean change in mass of potato pieces. Let's say you're taking 5 potato pieces, putting them first in a concentration of 0.2 moles per decimeter cube, then you measure the mass change after 1 hour, and then you repeat that experiment for different salt solutions. So that's the kind of a gist of the experiment. Now, whenever you're writing a method to investigate any experiment in a required practical, what is this testing? The required practical knowledge. Do you know the know-how of the scientific method? Do you know what are the control variables and the reliability? And the examiner's expectation here is, are you giving them a logical, a stepwise method, not just some random points written which will not score you the full marks? Now, to investigate this effect of salt concentration on the mass of potato pieces, the first thing you're going to tell is you're going to cut the potato cylinders, and they have to be identical to each other. So therefore, you're going to say 2 things about them: length and diameter. You're going to make them the same. So how are you going to make them the same? You're going to use a cork borer and ruler. Remember, so that you're measuring the length and you're making sure the cork borer is cutting all of the same diameter. Now, that's step one. Step two, you're going to measure and record. Just don't write measure. You have to use both these words, measure and record the initial mass. Yes, the initial mass of each potato piece. And how are you going to do that? By using a balance, a weighing balance. Now, 3 is you're going to prepare salt solutions. Yes, you have to prepare different solutions because each solution you are testing it at different concentrations. So therefore, you're going to prepare salt solutions and you're making sure they are of different concentrations. But remember, you are going to place equal volumes of each solution. Now remember, let's say you've cut 5 pieces of potato cylinders, then every salt solution of different concentration should have those 5 pieces. So and also equal volumes of salt solutions, you place them either onto beakers or test tubes. That's your choice. You can use either of them. Now you're going to place 1 potato piece into each solution and leave them for same amount of time. That's the next one. So you're going to take each of those 5 pieces, you're going to place 1 piece, potato piece. That is the minimum. You can go maximum how many ever pieces you want into each solution. But remember, it is recommended that you do it with 1 potato piece per solution, and you have to leave them for same amount of time. That is also important. The mass is same, the diameter of the potato is going to be the same, and the volume of the salt solution is going to be same, but varying strengths of concentration, and that is for same amount of time. And after that set time, you're going to remove those potato pieces. You're removing them. And then you're going to gently, blot them dry. You have to remove the excess solution. You're going to blot them so they can dry off. And then you're going to measure the final mass. Remember, you have to repeat the experiment for each concentration. Remember, you've done this. So you've taken those 5 pieces of potatoes, you've placed them, because every time you're calculating the mean mass, yes, of all those 5 pieces. Now, you have to repeat this experiment and make sure for each concentration, but remember, you have to calculate the mean change in the mass of all the 5 pieces. Yeah, that's why you're taking 5 pieces, let's say, and you're placing each potato piece into each solution. You're leaving them for the same amount of time, and then you are, after you take them out, you're blotting them dry and calculating the mean change in the mass. That's your 6 mark. Now, for this required practical, please write down the mnemonic SMCTRM, or the abbreviated shortcut. S for same size potato pieces. That is for S. M for measure mass before and after. C, changed concentrations. T, time kept the same. R, repeats in different strength of concentration. And M is the mean calculated. Now remember the examiner tips. You must mention the mass. You must mention the repeats. Writing do the experiment is not going to give you any marks in the mass of all the 5 pieces. Yeah, that's why you're taking 5 pieces, let's say, and you're placing each potato piece into each solution. You're leaving them for the same amount of time, and then you are, after you take them out, you're blotting them dry and calculating the mean change in the mass. That's your 6 mark. Now, For this required practical, please write down the mnemonic SMCTRM, or the abbreviated shortcut. S for same size potato pieces. That is for S. M for measure mass before and after. C, changed concentrations. T, time kept the same. R, repeats in different strength of concentration. And M is the mean calculated. Now remember the examiner tips. You must mention the mass. You must mention the repeats. Writing do the experiment is not going to give you any marks. So it's really important, the SMCTRM. If you want, pause the video and please go ahead and read that again. Write that down for osmosis required practical. SMCTRM. That is really important. Now let's move on to 5.2. Now this 5.2 and 5.3, you have to have a look at the graph. So this is again that nested question. Most of the questions in your GCSE papers are nested questions. What are nested questions? They'll give you one question, one set of data, and keep asking you multiple questions. Like in this, you've got question 5, which has 5.1, 5.2, 5.3, and so on, all based on that graph. Now, what is 5.2 saying? So we're looking at a 3-marker question. It is asking us, explain the result for potato pieces in the 0.6 moles per decimeter cube salt concentration. Now let's go back to the graph once. Okay, so at 0.6, you can see that here it's at negative 0.2 grams. It has lost. The mean change in mass of potato pieces was losing negative 0.2 grams of potato mass. Now, what has happened here? The potato has lost mass. And what is the mark scheme logic we need to apply? So there is osmosis happening. There is water potential which is involved, and there is the direction in which the water is traveling. Now let's break this down. At a salt concentration of 0.6 moles per decimeter cube, the solution has lower water potential than, that's a symbol for water potential, than the potato cells. So therefore, what it means, water is moving out. So water moving out of the potato cells. So when water is moving out of potato cells, and that is by using osmosis, and very important word here is partially permeable membrane. That's the important phrase here. Whenever you are describing osmosis, you have to write this. And this is causing the potato cells to lose mass. Sorry, not cells, the potato pieces. This is causing the potato pieces to lose mass. Now, as I said, it is important to write down 3 marks, 3 sentences. I'm going to repeat this. At a salt concentration of 0.6 moles per cubic decimeter, the solution has a lower water potential than potato cells. Water, therefore, is moving out of the potato cells by osmosis through the partially permeable cell membrane. This is causing the potato pieces to lose mass. Remember, lower water potential, water moves out, mass is lost. Higher water potential, water rushes in, mass is gained. Now, common mistakes I see as an examiner, writing salt moves, salt doesn't move, saying it is diffusion, rather than osmosis. Remember, diffusion and osmosis are exactly the same process, but water is special. In chemistry, I've explained to you why is water special. Water is special, it has its own name. So any movement of water either into the cell or out, we always call it osmosis. Very well done if you got that question right. Now let's move on to the last subsection of this nested question, question 5.3, which is again for 2 marks. Now let's read the question. Explain why the result for the potato pieces at 1 moles per decimeter cube was different from the result at 0.6. Now, the examiner focus here, and the command word is explain. They want you to evaluate what was the mass loss for at 1 moles per decimeter cube and at 0.6. Now, let's go back to the graph. We have already drawn a line here. We are going to draw the line here. As you can see, it is almost closer to, negative 0.29, almost negative 0.3 grams of mean mass loss. Now, here the examiner focus is on comparative explanation. Are you able to identify? Now, at 1 moles per decimeter cube salt concentration, it has even lower water potential than what we discussed at 0.6. So therefore, there is a larger water potential gradient. So the water potential gradient at 1 mole per decimeter cube is larger than that of 0.6 moles per decimeter cube. So therefore, there's more water moving out of the potato cells. That's very important to write down. The comparative explanation. You can also, to impress the examiner, talk how the mean change in mass was loss of negative 0.2 at 0.6 moles per decimeter cube compared to almost negative 0.3 grams of loss in mass. And you have to mention that at 1 moles per decimeter cube, it was a greater loss in the mass. That's the important mark clincher in this question. So comparison phrase, lower water potential, steeper gradient, more water movement. For this kind of questions, you have to compare. If you rewrite what you have wrote in question 5.2, then the maximum mark you're only going to get is 1. So wherever you have osmosis questions, do remember it's only the water which moves, not the salt solutions. And movement is always from high to low water potential. Remember the phrase partially permeable membrane in osmosis questions, and it's always osmosis whenever the water moves. It's never the diffusion. Now that's completing question 5, one of the most important questions in GCSE Biology Paper 1. If you can confidently answer this question, you're securing method marks, explanation marks, and comparison marks, exactly what examiners are looking for. If this video helped you, make sure you subscribe to my channel, dbtuition09 on YouTube. We have a presence on TikTok and Instagram as well. This is part of our full question by question AQA Paper 1 GCSE Biology June 2024, and it's designed to help all my fans understand how the marks are actually awarded from the examiner point of view. And I'll see you in the next video. Stay tuned.